Hello and welcome to Biological Structures and Roles of Proteins. I'm Dr. Mark Williamson for Transcend. This is a professional development core short video for the short video series. Let's get into it. So what I want to do with this short talk is talk about proteins, but specifically sort of proteins beyond sort of the basic stock understanding. Now, on one side, the more conventional lay conception of proteins is going to be from a nutrition label. When you think of protein, you probably think of like a slab of meat or some other protein. Now, if you're in a little more science bent, maybe thinking about introbiology, you probably have a more abstract understanding of protein from that view. Probably something with like the central dogma of molecular biology. You know that, well, DNA goes to RNA, goes to protein. And so it's sort of a very abstract thing here. But what I want to do here is really dig more into like what proteins are and their roles more broadly to help fill in the gaps between this macroscopic level and this microscopic abstraction. So that's what we're going to do here today. So let's start by defining a protein. Good description of that a protein would be that they are biological macromolecules comprised of amino acids. If you remember in introbiology, there's four major macromolecules. There's lipids, carbohydrates, nucleic acids, and then proteins. And yeah, they're made of smaller building blocks known as amino acids. Now, elementally, they are composed of carbon, hydrogen, oxygen, and nitrogen. And then for a few of the amino acids, they have sulfur and even one special case, selenium. So that's what they're actually made of at the elemental level. Now, the name protein, its etymology comes from the Greek proteos, means primary. And their role within the body, again, very abstract, but they have a wide number of functions inside living organisms. You can think of proteins as doing almost everything in the cell, even making up parts of the cell. But again, that's too abstract. So we want to get past that and get a fuller grasping of what proteins really are, what they really do. So it's good to talk about protein structure. And that's again, a very common thing you learn in nutribiology that there's different types of protein structure. Now, we already said that they're made out of amino acids, so that's their primary structure. What their actual chain of smaller molecules, those are known as amino acids, actually compose of. And we'll see more of the details of the amino acids later, but that's at their baseline. Now you can step up here. You have two different secondary structures, how so they start to interact. You can have these alpha helixes as well as beta pleated sheets. And taken together here, you start to get the three-dimensional shape in the tertiary structure. The protein will actually fold in certain combinations depending on that primary and secondary structure. There'll be things like hydrophobic interactions, hydrogen bonds, even ionic bonds, or even covalent bonding to form a complex, often complex three-dimensional shape. In fact, the idea of protein folding is a very tricky biological problem that we're only just getting a better handle on. And then finally, we have a quaternary structure. That's when often proteins are complexes of multiple sort of separate poses that come together to work together. And so that would be the quaternary structure, the higher order structure. You think of like hemoglobin has four components. That's a common example of quaternary structure. So proteins, fairly complex, have a lot of levels of structure you can analyze. So next, let's turn to those amino acids, so the primary structure. So there are 20 common amino acids across the tree of life, and all of them are going to have an amino, so that's the NH2, and then the carboxylic acid, so that's the OOH, and that's why they're called amino acids. They have an amino group and an acidic group. And then what differs is what they call the side chain, sometimes abbreviated as R. It can be very simple, like glycine. It can be fairly complex, like tryptophan. And the different side chains will confer different properties. You can see they're often separated out on different types of properties, whether they're negatively or positively charged, aromatic, amidic, etc. And in fact, some of them even have sulfur in them, like cysteine and methionine. Now, these are the 20 common ones. There's actually two more. That are found across certain organisms. These are sometimes known as kind of the alternatives. There is selenocysteine, which is very analogous to the more common cysteine, but in this case, selenium, the element selenium, takes the place of sulfur, and this is very rare. It occurs in specialized tRNA in bacteria and I think in some eukaryotes. Similarly, there's pyrrolysine, which is analogous to lysine, but has a different side chain. So lysine has a little bit simpler side chain. Pyrrolysine has a more complicated one. And those are found in methylogenic archaea and bacteria. Now, taken together, these 20 common and two alternative are sometimes called proteinogenic amino acids because they actually encode for proteins that actually make up proteins. However, that is not the entire space of amino acids. There are maybe what you would call non-standard or maybe not proteinogenic. And there can be over 100 of these, and they can occur naturally in different forms. And so this is a good conceptual space of amino acids by Matteo Ferla. You have all amino acids, and within the sort of the L stereoisomers, and we'll talk about that in a moment, you have, the proteinogenic ones. There are just 20 of them, but then there's other ones that can fall in there. And they can be these other amino acids that aren't proteinogenic. They can be intermediates at biosynthesis. They can be post-translational forms, actually made abiotically. They could be natural pharmacological compounds that don't actually make up proteins but have other functions. And of course, they can be man-made. Humans have created thousands of artificial, or sometimes known as non-canonical or unnatural amino acids. And another major point here, you can see the split of the all amino acids, the green slice, is actually, when it comes down to chirality, there are hypothetically D stereoisomers. So L stereoisomers is left-handed, so that's the chirality so the handedness of the molecule. Most amino acids are left-handed, but actually a few do occur in things like bacteria and antibiotics, the right-handed, the D stereoisomers. And then theoretically, I think from almost every regular amino acid, you can have the chiral inverse. So there's actually a fairly wide universe of amino acids that you could theoretically build proteins out of, though only a small subset actually make up current proteins. All right, so we talked about what they're built out of, a little bit more of that space. Let's talk about what they actually do, because that's fairly important for cell biology, important for living organisms. So I took this from my old cell biology textbook, a couple decades ago, still fascinating, of some broad broad categories of what they can do in the body and the cell. And I thought this was a good place to start. So first of all, there's enzymes. So for example, pepsin will break things down in the stomach. You have DNA polymerase. Again, enzymes more broadly are things that catalyze reactions. And a lot of times enzymes in the body are for like decomposition reactors. They cut things apart to help make things go apart faster, like DNA polymerase that helps with DNA replication. We have protein kinase that can add phosphorus groups to, I think, proteins post-translation. We have structural proteins. Some of these are macromolecularly obvious examples, like keratin, that's where hair and like horns get their strength. But then at the intercellular level, tubulin and actin are very common proteins. That tubulin makes microtubules, actin makes filaments to really provide structure inside the cell. Yeah, transport proteins, if you can think of things like serum albumin or hemoglobin, like transporting things like, again, hemoglobin like oxygen. Serum albumin transporting lipids, and those are almost like the trucks or the cars. And then, but there's also more like the roads or the toll booths. So there's like CA2 pumps. So there's proteins that form pumps in and out of the cell for various molecules. You got motor proteins like myosin. It's like in muscle. So when you think of a slab of meat, there we go. There's a lot of myosin in them. Kinesin and dynein are more inside the cell. Kinesin, I believe, helps move organelles around in the cell along like microtubules. Dynein, I believe, helps with flagella on cells. You got storage proteins, storing things like ferritin stores, as you might suppose, with the root like ferrous, like iron in the kidney, ovalatum and casein. Those are storage proteins in eggs and milk, respectively. Signaling proteins like insulin, other broader categories of signaling proteins would be hormones. Hormones are signal molecules. You have things like nerve growth factor, epidermal growth factor, and for every signal you often have a receptor. So receptor proteins like an insulin receptor for the insulin. Rhodopsin detects light, receives light. There's like a adrenergic receptor, for adrenaline, and then a huge group of proteins would be like all with gene regulation. So a common stock example in bacteria when you're learning about sort of genetics is like the lactoperon, and so you have a lactose repressor protein, and there's all sorts of homeodomain proteins, and really you can just think of like all the ways that gene regulation happens, especially in eukaryotes and higher organisms. You've got enhancers, repressors, silencers, all these huge protein complexes, either getting things like opening up the chromosome. And so there's a lot of moving parts, or you could spend your whole time, just studying that aspect there. And then there's even special purpose proteins that kind of don't fall in these other categories. Like there's Antarctic fish that have antifreeze proteins in their blood to keep from freezing. There's monolin, a sweet substance produced by some plants. And there's even glue proteins that organisms like mollusks or other invertebrates secrete to latch onto rocks even underwater. So a lot of different roles. Now, if you want to really dig down into even more specific roles, you can look at classifications from something like gene ontology, which is a structured, standardized representation of biological knowledge. Species agnostic, so you can look across the tree of life. It really enables consistent gene and gene product annotation, gene product being proteins, one of the products. And there's three aspects you can look at. You can look at cellular component, so where it is, biological process, and then molecular function. So here's gene ontology. So for cellular component, it's pretty sparse. But if we look at biological process, you can see there's a lot more categories. We have things like regulation, growth, pigmentation, even response to stimulus, all sorts of things. And then the real big one here is molecular function, also known as gene product or complex activity. Like you have things like binding, molecular carrier activity, protein folding chaperones, toxin activity, transport activity, catalytic activity, so enzymes. So you could really dig into these and the subcategories to look more and more at what proteins do and the different types of proteins. All right, with that, let's kind of close with some really interesting proteins, sort of the record-breaking or sort of champion-like proteins. The largest protein is titin. It is 27 to 35,000 amino acids, so it's huge, and it's used in muscle contractions. The smallest, or one of the smallest, is hard to the point where, like, because you can get kind of down. It's hard to tell like what becomes like a protein and just like a chain of amino acids. But this is, I think, the smallest folding protein that has an activity. It's tau pri. It's only 11 amino acids, and it's in Drosophila, so those are fruit flies, and required for development. The most abundant protein is collagen. It's in 25 to 35% of the proteins in a cell. So a fourth to a third of all proteins in a cell is going to be just collagen. It's the structural component of things like connective tissues, extracellular matrices. One of the most widespread proteins is called ubiquitin. That's where it gets its name. It's ubiquitous, found in all eukaryotic cells, and it has regulatory functions in things like tagging proteins for degradation. One of the most utilized, at least from a biologist side, is GFP, green fluorescent protein. It was derived from jellyfish. It glows green when fluoresced and can be used for visualization in molecular biology. And one of the first described, like more fully, was myoglobin. That's a protein that binds to oxygen and iron, and it's the first protein to have a 3D structure determined using X-ray crystallography. So with that was a little bit more about proteins, their structures, and their functions. Hopefully that helped fill in the gaps between a broad or abstract understanding in your mind about proteins. Thanks for joining me for this short video series. I'm Mark Williamson. I will see you next time.